So in Chapter 6, we finally get a chance to start talking about cells. And this chapter is titled A Tour of the Cell. A couple of things that we've already established, right? We know that all organisms are made out of cells. We also know that the cell is the simplest collection of matter that can still be alive, right? And cells can perform all of those functions of life that we discussed way back in Chapter 1. We also, in Chapter 1, talked about the unity and diversity of life, right? We understand that it's easy to find differences between different species of organisms on our planet, but maybe a little bit more difficult to do is to find some of those similarities. And yet cells definitely share many similar features irregardless of what organism you're looking at. And this will become a little bit more clear as we start talking about, well, what kinds of structures do cells share in common? Now, everything that we've learned about cells to date has depended on technology. We can't really see a cell using the naked eye. Maybe there's one cell, the frog egg, that's large enough to actually see using the naked eye, but you would pretty much see just a period on a page. You wouldn't be able to tell any sort of unique characteristics or features on the outside or inside of that cell. And so we've depended on the development of the microscope to help us visualize those very minuscule cells. Now, in this particular figure on this slide, we have essentially a spectrum of different sized objects that you might run into on a daily basis. On the left-hand side, you've got your really large objects, right? You've got a human. That's pretty easy to see with an unaided eye. We are fairly large. And then we go down to different size cells, and then all the way down to maybe bacterial cells, viruses, and then small molecules here towards the end. And you'll notice that for each sort of section of different sized objects, we also in this image show you how we could potentially visualize those objects. So with the unaided eye, we can see really large components all the way down to frog egg, maybe getting really close to seeing a human egg cell, but not quite getting us there. And so when we want to study cells, microscopy is really the best way to go. Now, there are two main types of microscopy, and we'll discuss both of these. They include the light microscope and the electron microscope. A couple of differences between these two, which we'll stress, but just initially looking at this slide, you can easily tell that the light microscope might be a little bit better at seeing larger components, cells, your average run-of-the-mill cell, can definitely be seen using a light microscope. Even some, bacterial cells, while you can't see a lot of their maybe unique structures, you can definitely see the cells using a light microscope. If you want to see anything smaller than that, right, you're trying to visualize maybe viruses or trying to see some smaller molecules, in this case, you're going to have to turn to the electron microscope, which has slightly better resolution and magnification for visualizing those small objects. There is a little other section here attached to the light microscope. This is called super resolution microscopy. We're not going to talk a lot about that, but there are some new techniques that scientists have come up with that pair essentially biochemical techniques with the use of a light microscope to help. Some are biochemical techniques, some are more technologically based techniques that allow you to use a light microscope, but you enhance the resolution and magnification, maybe more so resolution of whatever image it is that you're trying to see. It allows you to see smaller components than just a plain light microscope on its own. So let's talk about these two types of microscopy. So we've got the light microscope and the electron microscope, and we'll go into some details regarding both. But irregardless of what kind of microscopy you're looking at, there are three parameters of microscopy that are important to cover, and you'll also get probably a few more when you actually use the microscope in lab. And so in this slide, we are going to cover magnification, resolution, and then contrast. So magnification, this one is pretty easy, right? This is how much larger does the image appear under the microscope than it does in real life. So the ratio of an object's image size to its real size. Next, we've got resolution. On this slide, it defines resolution as the measure of clarity of the image or the minimum distance of two distinguishable points. So this can be kind of hard to comprehend. And maybe a little bit difficult to do when we're not in a classroom setting, but let's give it a shot. So let's say I drew 2 points, right? So I drew blue point over here, and another blue point over there, right? And I drew them pretty far apart. Probably, looking at your screen, you can see that those are definitely two similarly sized points that are fairly far apart. If you got up now and went to the other side of the room that you're in, you could probably still identify that this is two individual points. Now, let's try something else, right? So go ahead and walk to the other side of your room. All right, get up, walk over there, and don't look at the screen for a second. And I'm going to draw something on the screen that's below the original 2 points that I drew. Okay, so take a look at your screen, and hopefully you're far enough away where you can see that I drew something below the original 2 points, but what I drew looks maybe like a single larger point, right? You're far enough away where you can't tell that I actually did draw two similar sized points that are just really close together, right? Being far away from the screen, you don't have very good resolution. You can't differentiate that those are actually two individual points, when in fact they really are. And that's kind of what resolution is under the microscope, right? It's how well can you see really small components and see them sort of individually as opposed to just a blur under the microscope. And then lastly, we've got contrast. So contrast is pretty important in that it helps you visualize your sample. And sometimes you'll have to increase contrast, a couple of different ways to better identify your cells, structures within the cells, etc. But essentially, contrast is the visible difference between the brightness, in brightness between different parts of the sample. So that means how dark are your darks and how light are your lights. If everything's kind of a medium gray, it's going to be really hard to differentiate what's what. But if you've got really dark sections and really light sections, that difference in brightness will help you visualize your sample. Okay, so let's talk about the light microscope first. Essentially, a light microscope will use visible light. It'll pass the visible light through your sample and then through glass lenses, and these glass lenses are able to refract, or is called bend the light, so that the image is magnified. And so on this particular slide, we've got an example of what is called bright field light microscopy, where you simply use white light, you illuminate your sample, you collect that light, pass it through lenses, lenses magnify it, and you can see your sample. This is actually the same sample. These are cheek cells. But in the first sample that says unstained specimen, here we've just taken the plain cheek cells, put them on a slide, and looked at them under the microscope. And you can tell that there's not a lot of contrast, right? Everything is kind of the same color gray. Yeah, you can see probably the outlines of the cells, maybe the nucleus. Other than that, it's pretty hard to see much of anything else. And so in order to ease the viewing of this sample, we would add a dye. The dye would be picked up by the cells and it would increase the contrast. And that's what you see on the right-hand side with a stained specimen, right? So those are the same cheek cells, maybe a different part of the slide, but same principle. And you can see that, wow, the nucleus is that really dark black spot in the middle, and then the outside of the cell is really bright and white. So you have great contrast and you can see a lot more detail within the cells. Now, there's another kind of light microscopy besides bright field microscopy, and this is called fluorescence microscopy. Instead of using white light to pass through your sample, fluorescence microscopy uses fluorescent dyes or probes to label different parts of the cell. So number one, this is really beautiful. This is a fibroblast cell, and you can see that there are three colors, right? So there's a fluorescent dye called DAPI that stains DNA. So you can see the blue nucleus where the DAPI and the DNA are located. And then we've got two other probes that were labeled with a fluorescent marker. One's red and one's green, and you can see that they also stain different parts of the cell. So in fluorescence light microscopy, you can get a lot more specific. If you use a specific stain or probe, you can label a certain protein or a certain structure within the cell, giving you that added information when you're looking under the microscope. But again, it uses fluorescence instead of just regular white light. Now, the cool thing about light microscopy is that the way that you prepare your sample, you can actually maintain a living sample under the microscope. So you can see things swimming under the microscope. You can see life happening at that microscopic level, which is definitely very cool. The downside to a light microscope is, it doesn't have that good of magnification. So there's limits to how small of objects you can actually clearly visualize using a light microscope. If you want to try to see those very small objects, getting down to smaller molecules, you'd have to use an electron microscope. Now, electron microscopes, instead of using a beam of light to pass through your sample to visualize it, they actually use a beam of electrons to bounce off of or pass through your sample, and then the electrons are collected on the other side of a sample using a detector, and then the detector sends an image to the computer screen, and it shows you a created image based on where the electrons landed on the detector. Downside to using a microscope, an electron microscope, while you can see very small things, you have to kill your sample. So typically the sample will be prepared in such a way that it's very, very thin, and then it's coated with a heavy metal. And so this doesn't allow for life to continue at that microscopic level. When you coat the specimen with the heavy metal, it allows the electrons, as they are pointed towards the sample, to bounce off of the sample, right? So the electrons will hit the heavy metals, they'll bounce off in an array of different patterns that's based on the structures that are present within the sample. And so the electrons, based on how they bounce off, the detector will collect them and create an image. Now, there are two different kinds of electron microscopes. So on the top here, you've got what is called a scanning electron microscope, or an SEM. You can think of this as a sonar, right? A submarine has a sonar, and it can map the topography of the bottom of the ocean, so you can see all the hills and the valleys. Scanning electron microscopes do just that. They focus the beam of electrons on the surface of the specimen, and they scan the surface, so it gives you a three-dimensional image of the surface of that sample. And so on the right-hand side, this is an image taken using a scanning electron microscope of the surface of the cells that line your small intestine. Now, the surface of those cells is covered in something that are called microvilli, tiny little projections, smaller than cilia. And they don't move. All they do is increase the surface area of those small intestinal cells so that you can absorb a lot of nutrients as the digested food passes through the small intestine. And so you can see all those little microvilli here on the surface of those cells. Now, let's say we wanted to see not the surface of our specimen, but we wanted to see inside of a cell. To do this, we would end up using a different kind of electron microscope that's called a transmission electron microscope, or a TEM. These guys will actually focus the beam of electrons directly through the specimen, and so you get some information from the inside. So if you take a look at the image here in the lower right-hand corner, these are the same small intestinal cells. Only difference is, instead of allowing the electrons to bounce off the surface of those microvilli, now the electrons are passing straight through. So you can see some cross sections through the microvilli. You can see some of the internal fibers that give the microvilli their structure there.